Recording. So again, good morning, everybody. And I'm reminding you that these recordings are always posted after the class. If you have problem in accessing those on Zoom, because most of the time on the cloud, I also created those on a YouTube channel. It's only locked for you guys, and then go and have a look on the classes. So, okay. So today's lecture is mostly about cell death, and we will be discussing part of the metabolic diseases, okay? As you can see in the introduction slide, I added this scheme here. It's very important to remember. So this scheme is showing you the concentrations of calcium outside of the cell and inside the cell. I'm assuming you have got that in different classes, like cell biology probably, biochemistry as well. So the aspects of having
Excuse me, Professor. You're not sharing your screen.
okay. Okay. Thanks for I okay. Okay. Can you see it now?
Yep.
Could you? Yeah. Okay. Thank you. Okay. So technology. Well, thanks for reminding me, because many times this happened before and I was like on an online class and the slide doesn't show and I speak and nobody says anything. So thanks for poking me if this happens. So I said in the beginning of this class, I have this slide here, which is showing you the levels of calcium, concentrations of calcium inside the cell and outside of the cell. So this is an aspect that we really have to remember, because it tells us a lot about the function of calcium in the cell and how it's organized. And it also gives us an idea that if for any reason this homeostasis is broken, where the levels of calcium become high, this can be detrimental for cells. Okay. So in the previous lecture, we have focused on the metabolic pathways, oxidative stress, and the generation of ATP from these nutrients. In this lecture, we will relate into mitochondrial damage. We will focus on the calcium concentration and what happened to calcium and how that could be a trigger for cell death, and then for therefore some diseases. One of them is diabetes. Office hours are still on the time. Some of you are requesting to change these. There's only few. Last week I didn't have anybody during the office hours. Please start digesting the material as soon as possible. So today we are going to connect the metabolic dysfunction to the disease states, and particularly the loss of ATP. We will continue about the mitochondrial damage and get an overview how that happens. One of the aspects that we will discuss is the levels of calcium, as I said, and its normal function and its function during the damage. We will discuss cell signaling pathways, events that differentiate the different types of cell death. And the way I'm going through this different types of cell death is not really just to state them. It will be mentioned throughout the slides. Like if we have a type of cell death that is related to a particular event, then we will describe it in a slide next to it. And finally, the type 2 diabetes. I hope we'll have time to get to that point. So one thing I would like you to do is really start interacting more during these classes. So I'm opening the discussion after the break to have questions related to what I'm discussing and to have suggestions from you on aspects that you think it was not explained enough. So the component of the discussion will be regular in the next coming classes. It will be like around 20 minutes in each class. So you remember this, probably this was the last slide of the class of the previous module. And we discussed the adaptation process after injury that could be translated into different aspects of how the cell behaves. If we take these two, the easiest one here, the atrophy and the hypertrophy, is that the cell probably shrinks because of the lack of nutrient or oxygen or ATP or due to a toxin or a drug. And at this one, the cell goes opposite. It's hypertrophying, meaning the cell is getting bigger. And those situations may happen in the same tissue. Like, for example, atrophy and hypertrophy are aspects of the cardiac failure, uh, where hypertrophy is an increase in the cell size because we are losing a lot of cells of the heart at certain point of stress, and then those surviving heart cells become bigger in order to keep pumping the blood toward the organs. Certain drugs that we use sometimes cause atrophy, and this is very well known. So these this is always a comment I'm saying that yes, we are kind of dismantling those pathways, and it looks like these pathways are separate, and it looks like they don't happen in the same tissue or organ. Please bear in mind, this is not the case. Many of these cellular aspects, signalings, uh, molecules, signaling molecules are taking place within the same tissue. Now, if the adaptation process was not enough, or in other words, if the stress was higher, or if the damage is uh, uh, in a magnitude that the cell has to repair, then we can go to irreversible versus an irreversible uh, aspect of repair. The reversible aspect is that if we have a loss of ATP, we will recover that. If the cell swell is like already swelling, then we can get rid of the water. If the ribosomes have detached from the sarcoplasmic reticulum, those will come back and reattach. The autophagy is a part of that because we can get rid of all these components that are damaged. However, and this brings us to the update where we are in the class, the mitochondrial vascularization, which is, in principle, the mitochondria is not going to be functional anymore. And one of the component that is important on top of that is the calcium influx. Okay, and at this point, This is a point of no return. Now, the types of the cell death, yes, we characterize them many types of cell death, but at the end they have very similar aspects that are common. The factors that classify one cell death compared to another is like kind of different, but they have a lot of common aspects as well. So the first thing we have to follow up is the mitochondrial damage. So we discussed in the previous class how the mitochondria use all these nutrients to generate the energy. Now, while the mitochondria is using that oxygen that we breathe and maintain that H plus gradient across the membrane in order to pump it and generate ATP. So we went extensively on that. So I'm not going to go back to repeat it. One of the side products that is generated is the reactive oxygen species, which ROS, which today I will revisit again a few of the slides from the previous classes to reassure that we have a very good information about how reactive oxygen species affect cellular function. And in mitochondrial, like in terms of mitochondrial damage, there are other aspects. Some of these are genetic, for example. I'm going to focus in this class on the levels of calcium and the ROS. So what happens when there is a mitochondrial damage? So the event that really there is no comeback is the loss of the membrane potential. And therefore, the integrity of the membrane is broken. And if you remember from that video that I showed you how the electron transport chain, this is a very, very important video. I really like it, and I really uh, uh, would like you to watch it again and again whenever you study for the mitochondria, so that you remember how these complexes are functioning and how they are placed and how the electrons are being transported from one to another. So once the membrane potential is broken, or the integrity of the membrane is broken, which is the same, then the direct effect would be the depletion of the ATP. So you will have a massive loss of the currency of energy in the cell. Okay. And eventually, there is cell death that most of the time it goes through the necrosis, which is a type of cell death that is not organized. Okay. However, some of the other proteins that we discussed is the cytochrome C. Remember the cytochrome C, which is responsible for the transport of some of these electrons from one complex to another. And cytochrome C is residing within the mitochondrial inner membrane. So when there is a damage in the mitochondrial outer membrane, then cytochrome C can be released. So the moment that a protein that is normally found within the mitochondria is released to the cytoplasm, then this will trigger a signaling pathway that will be sensed that, oh, the mitochondria has some damage, because I am now able to see cytochrome C, which is normally supposed to be within the mitochondria, is in the cytoplasm. So what's the reason? And cells will directly act on that. Okay, so we will see how that happens. In terms of the types of cell death, it's not strictly always necrosis, where the cell permeability is the cause of that necrotic cell death and the loss of the membrane potential, and therefore there's not a lot of ATP that's being generated. Mitochondria also converges at the apoptotic pathway, which is an ATP-dependent type of cell death. because of a pro-apoptotic proteins being released, and one of them is the cytochrome C. So if I ask you a question, mitochondrial damage cause necrosis or apoptosis? Yes, it causes both, depending on the trigger and depending on the physiological state that caused what to happen. And we will be describing few scenarios today to explain to you how we differentiate between these two types of cell death. So the other aspect is the increase of the cytosolic calcium. Okay, well, the first slide that I showed telling you that the levels of the calcium 0.1 micromolar. Be aware, 0.1 micromolar within the cell. Outside the cell, 1.25 millimolar. You are speaking about 1,000-fold, 10,000-fold higher. Okay, so that's a huge number. And therefore, the fluctuation of the cytosolic calcium would definitely be a trigger for a cell-mediated injury, adaptation, and therefore most of the time cell death. So let's talk a little bit about cytochrome C. So cytochrome C is one of these proteins that it's localized here in this slide. And you will see in this lecture that I didn't really depend much on the textbooks. I depend more on recent papers because these data could change today and tomorrow or the day after or yesterday. Uh, so most of these references are coming from research papers. Yes, sometimes it's more complicated for you. I try to simplify and take the snapshot from this paper. Sometimes it's more complicated. I will mention what you need to know and what you don't need to know. So remember the complexes 1, 2, 3, and 4, and the electron transport between the complex 3 and 4 is mainly mediated by cytochrome C. Okay. Now, cytochrome C being residing in the inner membrane space, whenever there is an issue in terms of the mitochondrial membrane integrity, then cytochrome C will be released. Okay. So the cytochrome C mobilization involves mitochondrial cristae remodeling. Like we like to draw things nicely, so it's drawn like a straight line here. Obviously, the mitochondria is not like that. So these are the cristae of the mitochondria. So it's like really compact. And this is something important to know that D complexes you will have much more complexes 1, 2, 3, 4, or electron transport chains that are localized within a cristae that is organized this way compared to a straight line. So when the cytochrome C is released, then that's a trigger. But what keeps cytochrome C residing within the mitochondria? Which brings us to an association of this protein with the phospholipids of the membrane. So most of the cytochrome C, you can see it's very nicely drawn here. This is cytochrome C. It is anchored with the phospholipids of the mitochondrial membrane. And the name of these phospholipids is cardiolipin. Cardiolipin. Okay. Now, whenever there is an oxidative stress, and we will be describing what is an oxidative stress, that affects the phospholipids of the membrane. So you can tell now that the trigger has to affect the phospholipids. Then those lipids, most probably in this case, are oxidized. And you can see there's an OH group that is added here. then cytochrome C is going to be released. There is something that changed in terms of this curvature, and that's what we exactly speaking about. It involves remodeling of the cristae. So this curvature of the membrane of the mitochondria has been changed to a more straight line-like. And therefore, these dentations, you can imagine, now cannot hold the cytochrome C, and that's why it's released from the mitochondria. So this is one of the aspects of triggering cell death, and we will come back to this at the end when we describe apoptotic signaling. So what's the relation between the calcium load and the mitochondrial function? So this is real data, in fact, where it shows based on experiments that the activation level of the generation of ATP is strictly depending on the levels of calcium, because the physiological function of those mitochondria, where they could generate the maximum amount of ATP, here is the level of how much ATP you generate, is depending on the healthiness of these cristae, the levels of the calcium in the mitochondria or in the cell. Okay? I will be describing a little bit what's the difference between the two. But now, if the calcium load increases, so the moment you start to have an increased level of calcium, then you can start seeing reorganization of the cristae of the mitochondria. You can see that now these dentations here, you have many of them, you lost many, and now the mitochondria somehow swell a bit. And this dramatically affects the levels of the ATP because you can tell that there is a leakage of the proton. Now, if the levels of the calcium increase drastically, then this becomes not only problematic to the generation of the ATP, it will become an ATP degrading step. So you start losing ATP in order to maintain the mitochondrial integrity. So the levels of calcium is directly correlated to the healthiness of the mitochondria. And in other words, is affecting the powerhouse of the mitochondria, of the cells. So any fluctuation that is affecting the calcium level would eventually cause mitochondrial dysfunction. And you can tell if ATP is not generated, the cell is gonna most probably die. Which brings us back to this slide again. Yes, calcium plays an important and central role in many functions, including like muscle contraction, neurotransmission, the proliferation of a cell, the regulation of genes, their transcription, differentiation of cells from one type to another, metabolic aspects, transduction of signals, and also plays an important role in triggering cell death. So whenever calcium enters the cell, it is chelated or absorbed and stored within mitochondria, within this endoplasmic reticulum, and within effector proteins or buffers. So it's chelated within those three different components. That's how we know how much calcium we have. And if we need calcium, then we can get it from those reserves. And if we have enough and we don't need more, we can simply export it. And that's why we have exchangers here. You have two exchangers which bring the calcium out. So in the next slide, it's a little bit more elaborating on the calcium. So calcium levels, as I said, relates to many types of injury. Today, probably I will describe only ischemia, which is loss of oxygen in our tissues. This happens in the brain, for example. This happens in the heart. And why I'm saying brain and heart, of course, it happens on any tissues, because brain and heart cannot survive a long time of ischemia or loss of oxygen. And even if the oxygen has been restored, which we call reperfusion, this can cause damage. So if we have a loss of oxygen or ischemia, or certain toxins affect the membranes, this can cause an increase of the calcium in the intracellular level. So the smooth endoplasmic reticulum, where the calcium is stored, or the mitochondria, those, when they are affected by whatever triggers, toxins, ischemia, or inflammatory responses, then calcium level becomes higher. When the calcium levels are higher, we have multiple signaling pathways that take place in the cell. So be careful for the word intracellular. Between one cell to another, it's intercellular. Within the cell, it's intracellular. Okay? So calcium causes intracellular damage. This means within the cell, mitochondria, sarcoplasmic reticulum will be damaged. And a number of enzymes which degrade cell structure and component are now activated. So calcium itself can trigger cellular enzyme activities. For example, ATPase. ATPase digests the ATP, right? You have phospholipase. Again, I always try to read the name and think what it will do. Phospholipase. The ASE is a breaking enzyme, phospholipase, so it's going to break the phospholipids of the membranes. Proteases. Protease is an enzyme that breaks down proteins. Endonucleases. These are enzymes that break down DNA. Okay, so the proteases, I'm just going to give you an example here, could break the cytoskeleton. So all actin filaments, which keep the integrity of the cell so it doesn't collapse, and the contractility and all that. Imagine those are broken. Phospholipases, membrane damage. Endonucleases, DNA and chromatin degradation. And ATPase cause the depletion. So you can imagine this signaling event is already triggering by now 4 different aspects that each one of those is really a major trigger of cell death. So if we look at calcium, how does it enter to the cell and what happened to it? Just to reinforce this slide here, because it's not really very well explained. So you can see that calcium enters the cell through calcium channels. So it's very well gated, right? So calcium enters the cell through calcium channels. Once inside the cytoplasm, it has to directly be chelated by proteins. So certain proteins bind to calcium and sequester it. Okay? Then the organelles, the sarcoplasmic reticulum and the mitochondria, they store the calcium. And I want you to remember this name, sarcoendoplasmic reticulum calcium ATPase, which is SERCA. The short name is SERCA. So this is an ATP dependent Oh, alarm. So this is an ATP dependent pump for the calcium to the sarcoplasmic reticulum. So you consume energy in order to store this calcium in the sarcoplasmic reticulum. Okay. Now, let's imagine now that we have enough ATP and we have enough calcium, but now we need to exchange this calcium. So what do we do? These are the two aspects that are not well explained in this slide here. So we will have the sodium calcium exchanger and the sodium potassium exchanger. And so the sodium calcium exchanger and the sodium calcium potassium exchanger, those will pump calcium outside of the cell. Now, in conditions of electroporation, which means there's a loss of an integrity of the membrane of the cell, then the calcium will rush into that cell and leads to very high levels of calcium within the cell. Now, there is a YouTube here that describes for you the induction of calcium. At your convenience, go and watch it. It gives you an idea about how the calcium levels change within our body. And bear in mind always the magnitude of difference between the calcium within the cell and outside of the cell. This is a very, very important aspect you have to remember. Okay, so I kind of touched a little bit about the ischemia reperfusion injury. And this will come back to this slide two times. One time when we are describing the mitochondrial damage and calcium, and one time when we discuss about the reactive oxygen species. So what are the two scenarios that we are seeing in this slide? The first one is, this is a blood vessel, and the oxygen within the red blood cells is not being transmitted anywhere after this is thrombus because it blocks the blood flow. Okay. Anoxia is a total loss of the oxygen, and ischemia is a reduction of that amount of oxygen. So you still have some hypoxic condition that is not 100% blocked, like anoxia. So what happens there? Most of the time, the cells become swollen. And now I will describe for you what causes the cell to be swollen. Now, after intervenience, they moved. So the oxygen-containing red blood cells will now rush into that area that was deprived of oxygen. But the problem is, once we push that oxygen into the area, it will start generating a lot of hydroxy radicals, like superoxide, peroxide, and hydroxyl groups, which now can cause the cell to die by a necrotic cell death. So what causes that cell to swollen when there is no oxygen? So you can quickly relate to what we have discussed so far. And if you remember, we need that oxygen to generate the ATP. Without oxygen, we cannot generate ATP. So practically, there is a reduction on the level of the ATP that is being generated. And if we go back to what that ATP level correlates to, it correlates to the sodium-potassium pump, which is the sodium-calcium exchanger that we just explained here. This is one, and that's another. So you are not able to store more oxygen, and you are not able to pump oxygen out, sorry, calcium. I meant to say calcium, not oxygen. And you're not pumping this calcium out of the cell. So what happens next? The levels of intracellular calcium and sodium increases. So that's an increase of the level of these cations. And normally what happens is that the water is going to freely diffuse into the cell, and the water is following these cations because there's a demand for this oxygen, water to enter, because whatever there is a solute that is concentrated within our cells, the water follows. And this practically causes the increase of the amount of water within the cell. And eventually the cell will swell. But the swelling comes with effects that we just mentioned in the beginning, that now the sarcoplasmic reticulum will swell. And if there are ribosomes that are attached to the rough endoplasmic reticulum, these will detach. So you automatically block the protein synthesis at the level of the translation. The other thing is that we describe it there, which is the mitochondrial swelling will drastically cause more decline in the levels of the ATP. And therefore, now the mitochondrial integrity is at risk. So this dilation causes the ribosomal detachment, endoplasmic reticulum, and from the endoplasmic reticulum, and the integrity loss of the mitochondria. And therefore, at this point, it's a very critical point that probably this cell has to quickly act to block the swelling that is continuous at this point, or otherwise it's gonna die. Okay, so you can relate the levels of calcium to the increase of the size of the cell that is continuous. So the cell has to quickly adapt to that, block it in order to go back to normal, and otherwise this cell is dying. Okay, so the other aspect that is mentioned here is that, okay, now we performed the reperfusion. In other words, we removed this thrombus, and then the oxygen rush in with those blood cells. But is this enough to save the cells? In terms of ATP, we have the requirement, which is the oxygen, right? We have the oxygen back. So in principle, mitochondria, even in this swelling, is still coming back to produce more ATP. So try to restore the levels of the ATP, okay? The issue that at this point, we have another player that comes to the show, which normally the levels of the reactive oxygen species are very low, but now they become very high. So we have another imminent threat to the cell that we have to deal with, which is the reactive oxygen species. Okay, is this clear? Because I want to stop here for a minute before I move to the discussion of the reactive oxygen species and their effect. Is there any questions? Anything you want me to repeat? Okay, sounds good. I didn't get any messages. Okay. So the next aspect is the reactive oxygen species. The reactive oxygen species, we discussed this in module 3, 4, I think. And we described at those, there's like multiple slides that I just put here to remind you about. We have already discussed that, and if you have notes, go back and discuss those slides. That our cells, because we are using oxygen, there's no way that our cells do not produce reactive oxygen species. But then there are systems that are implemented in our cells that takes care of the moderate or low level of reactive oxygen species in our cells. Particularly, we described the NADPH, which is the major producer of these oxide ions, superoxide dismutase, which converts these superoxide to hydroxides, the pyridoxine, the glutathione peroxidase, the glutathione reductase, and the glutathione disulfide. So go back to these slides and really get really good grasp on what is doing each of those steps, because it's important for you at this point to start relating reactive oxygen species to mitochondrial damage, membrane integrity, and cell death. And you will see this in the next few slides. So here, we mentioned that we have enzymatic reactions and non-enzymatic reactions, and these non-enzymatic reactions are mostly provided by vitamins. So that's why it's important to keep those vitamins level on check. So how reactive oxygen species, my computer froze. Okay. So how reactive oxygen species cause the damage of the mitochondria, and what reactions are taking place in terms of removing the superoxide, the hydrogen peroxide. You can go to the book here in the pages 63 to 67. I took this figure there. Some of the text is written here for you because we already discussed this before. But just to summarize, for the sake of today's lecture, that whether radiations, chemicals, inflammation, or reperfusion injury took place, then superoxides are generated. Within mitochondria, there is the superoxide dismutase, which converts a superoxide to hydrogen peroxide, which is H2O2. So this is a less potent reactive oxygen species. And then the hydrogen peroxide could be dealt with the Phantom reaction, which involves metals like iron and copper to convert that into water, hydroxy radical and then water. Or it is involved enzymes like glutathione peroxidase that converts the peroxide into glutathione. So we have here the end process. If this doesn't happen, it will cause disruption of the membrane, protein destruction, loss and misfolding, and DNA mutations and breaks. We described this, but I will show you in this lecture the plasma membrane and the protein destruction. So the removal, it's written here for you. I really want you to remember this very well. The superoxide dismutase, the glutathione peroxidase, and the catalase. Okay, I want you to remember very well this one. So reactive oxygen species could be generated endogenously or from exogenous sources. Again, we described this. We extensively went to how that affects the transition from one base to another in this case from cytosine to uracil. We also discussed how that can break the DNA into single or double-stranded breaks. And also we described it extensively how we try to repair those breaks after reactive oxygen species or stress that causes DNA damage. What we didn't describe is the induction of cell death and lipid peroxidation. Okay, so here we are coming for almost 50 minutes. I will go for like 5 more minutes probably and take a break, and then we come back to discuss the rest. So reactive oxygen species cause destruction of cell membrane and cell structure. As I said, there is always reactive oxygen species that are being generated. And some of them, they play a role, in fact, in keeping the cell in homeostatic physiological state. Now, when the levels of the reactive oxygen species become so high, that's when those become damaging agents and become disease-causing agents. So peroxisomes generate reactive oxygen species. Mitochondria, we described this extensively in the electron transport chain, how it generates reactive oxygen species. Metabolic enzyme, when they convert one of these metabolites to another, they could generate reactive oxygen species. ER stress, which means there's a lot of protein that are being translated in the ER, it causes the ER stress. And this enzyme, the NADPH oxidase, which resides on the membrane, it's listed here in this slide as one of the proteins that causes the induction of reactive oxygen species. So all those cause the level of the reactive oxygen species to become higher. Now, reactive oxygen species causes DNA damage, cell lysis or autophagy or apoptosis, and then lipid peroxidation. So that's what we will discuss in the next few slides, the lipid peroxidation. So what is first an oxidative stress? So oxidative stress is induced by external stimuli, radiation, like drugs, inflammatory responses, or by the intracellular uh, signaling that I just described for you. Now, when the production of reactive oxygen species exceeds the capacity of the reductive rheostat, the mammalian cells experience a series of oxidative damages, and these we call oxidative stress. So in other words, when this system here, the amount of vitamins and those enzymes, these are the reductive rheostat. As much as we are generating of reactive oxygen species like superoxide, peroxide, hydroxyl radicals, and as much we have enough of the superoxide dismutase, or if we go there, the superoxide dismutase, the glutathione peroxidase, and the catalases, in addition to the necessary minerals or metals, then the reductive rheostat will be able to get rid of all these reactive oxygen species, and therefore this will not happen. The disruption and of the plasma membrane and the proteins and the DNA breaks will not take place. The moment that the reductive rheostat is exceeded by the levels of the reactive oxygen species, that's when the oxidative stress starts. So that's exactly what this sentence is telling you here. Okay? So the first aspect is that DNA damage can take place, and we discussed what are the molecules that are affected by DNA damage, and we will remind you about it, which is the p53, for example. The second aspect is the lipid peroxidation. So let's move to lipid peroxidation. So lipids, if I remind you how it looks like, these are like phospholipids of the membrane. So you remember you have a phosphate group and you have an saturated and unsaturated fatty acids. And that's a triglyceride, and this is something it's in our blood. So we have a glycerol with a 3 fatty acid attached to it. So remember this from the beginning of the class. So now these phospholipids maintain a double layer to generate our membrane. And within this membrane, there are proteins and glycoproteins that are attached. So it's very simplified here. It shows only the membrane of the phospholipids of the membrane. Now, when these superoxides are generated under the oxidative stress conditions that I mentioned, those free radicals attack lipids. And if I go back one more, so these are huge molecules. We have a lot of carbons here, and these carbons in the unsaturated part, the fatty acid, they have here the double bonds, right? And it kind of have this stoichiometry that is different from the saturated fatty acids. So it gives that small curve here that is presented. But at the same time, this is a target for having an attack by the superoxides, stealing the electrons from this unsaturated fatty acid. And that's exactly the trigger point that causes lipid peroxidation. So the free radicals attack lipids, initiating free radical chain reactions with molecular oxygen, a process called lipid peroxidation. Meaning, what you are saying peroxidation, this means it's oxidized and again oxidized. So it's more than one time oxidized. Lipid peroxidation has been closely associated with many diseases. I have references here for you, like all degenerative diseases probably are correlated with that. But what exactly causes the damage? So the lipid peroxidation mainly happens at the membrane incorporated polyunsaturated fatty acids. So these molecules here. And this type of the type of cell death that is really induced by this, it's called ferroptosis. And the first part of the name, it says ferro, it's iron related. So in the presence of iron, there is a type of cell death that is very similar to apoptosis that takes place. So what happens at the first step of the lipid peroxidation? It is that superoxide, hydroxide, or reactive oxygen species will make this reaction to take a hydrogen atom, so a hydrogen atom transfer, which is HAT, it's written here, or the peroxyl radical addition, or you add that to the side of the lipid. So what does this cause? Obviously, the reorganization of those phospholipids is going to change. And this causes the cleavage of those unsaturated fatty acids. Now, when these cleave, they become free electrophiles because they are now ready to go and try to regain back those electrons they lost. Because in principle, the superoxide attacked those unsaturated fatty acids and took some of the electrons, and now those electrophiles are gonna go and search to get those electrons back by doing the same. So now you have first affected the integrity of the membrane because this membrane is definitely healthier than this membrane. And second, you have these electrophiles ready to go and damage other components of the cell, like proteins and other lipids. Okay? So if I go one slide forward, there's a very simplified version of the lipid peroxidation and how they can cause the propagation of this process more and more. So this is the unsaturated part of a fatty acid. And again, I always try to simplify. There is a carbon which has one, two, three, and double like the bonds. One of these is a hydrogen. So this is an easy attack for the hydroxyl radical. So what did the hydroxyl radical do? They took that hydrogen away, right? So they took that hydrogen away, leaving this with no electrons. And now this lipid radical is gonna go and start searching for electrons. Now, in the presence of oxygen, this lipid radical becomes a peroxyl radical. And again, this is gonna try to find those electron because now it's in a transitional state where it's probably like one of these electrophiles. So you have this electrophile. So this electrophile could attack another unsaturated fatty acid, which is exactly the same like here. And this process goes again and again and again. And if you see the driver is hydroxyl radical, which is a reactive oxygen species that has been upgraded, like the levels are very high. And then we have oxygen, which is available. So as much as we have reactive oxygen species and oxygen, then the first step of initiation will cause this to happen to a lipid peroxide radical. And then this will go on another cycle and another cycle and another cycle. Okay? So that's how lipid peroxidation causes the damage to the membrane. Okay? So I will stop here. We'll take a break for, let's say, 20 minutes. And then we'll come back to finish up on the lipid peroxidation and move to the types of cell data. Uh, within After the break, we will open discussion a bit, and then we will resume the slideshow. Okay, so let me pause the recording. Can you see my screen? Okay, sounds good. So we described lipid peroxidation and what are the molecular events that's taken place. And then we move to describe what's the side products and how those affect different component in the cell, like proteins and the membrane. So as you can see, the type of cell death that I mentioned is the ferroptosis. So it's fairly depending on the levels of the iron. It is also ATP related type of cell death. You know, with time, if you now go and search for the types of cell death, a few years ago, there was apoptosis and necrosis, then apoptosis, necrosis, and necroptosis. Now you have ferroptosis, pyroptosis. There's lots of these categories, and this is a very growing domain. But the bottom line, they all converge at certain point. Yes, there is a particular aspect of the induction of the cell death, but later on, the cascade of the event is very similar. So ferroptosis, just to mention, this is very well dependent on the level of the antioxidants. So in other words, if the glutathione is not produced in the cell by the enzyme glutathione peroxidase 4, the moment that this enzyme fails to produce the glutathione, then ferroptosis takes place. Now, the trigger why the cell is not producing enough, or this is mutated, or the levels are not adequate enough of this enzyme, this is a different aspect. We're not describing it here. But it's important for you to know that this is depending on the nutrient availability. So particularly glutamate, which is transported and forms the cysteine, which is the precursor for the glutathione. So in other words, if the cysteine levels or cysteine amino acid level are low, which it could be a reason that the glutamate transport is not taking place, then this automatically feeds into a loss of the level of the uh, glutathione in the cell. This is one scenario. The other scenario is that you eventually have that route where you have enough cysteine and cysteine. However, the enzyme itself is not able to produce the glutathione. And in both situations, the result is the same, that loss of the glutathione is a reduction of the reductive rheostat of the cell, and therefore it's not being able to cope with the reactive oxygen species being produced, and therefore lipid peroxidation takes place and ferroptosis takes place. And if you see the conversion between the two arms of this type of cell death, is that for that to happen, lipid peroxidation increases and levels of iron is high. Okay. Most of these information are written here for you if you want to read. Okay. So we described how lipid peroxidation takes place, and we mentioned that there are few steps. In the next slide, we will describe the whole steps of the lipid peroxidation. So lipid peroxidation is like, it's similar to what described here, but it's summarized better in this slide here. So the first step is that reactive oxygen species are going to affect polyunsaturated fats, polyunsaturated fatty acids, okay? Those will generate the lipid radicals, the one we just showed here, the lipid radical, this guy. The next step, those will react with the molecular oxygen that is available to generate the lipid peroxyl radicals. Now those become the reactive molecules, which is represented here as this one. So this is the lipid peroxyl radical, where you have two oxygen molecules attached to that carbon on the unsaturated fat, or unsaturated fatty acid. Now, in the next step, this becomes the electrophile that can go and affect the unsaturated lipids. So in this slide here, you can see that now this guy can affect the PUFAs, which are represented here for you. So similar. So it attacks the PUFAs, and now you generate the lipid hydroperoxide, which is this guy here, the lipid peroxide, where you have OOH attached. Great. Lipid peroxide is presented. So we have the initiation step there, the propagation step where lipid peroxide radicals are being generated, and then the end product, the hydroperoxide. Now, those can become unstable, okay, and then can break down into different molecules. The 4-HNE hydroxy 2 neonal and the malonaldehyde, the MDA, these are the two most known breakdown aldehydes from those lipid peroxidase. And those are reactive species that could eventually perform the bad part of lipid peroxidation. So is this a reversible step? Is this something reversible which we can stop? Yes. As I said, it depends on the levels of the reactive oxygen species, because if there are enough antioxidants or enzymes, then the antioxidant can block these steps and then can regain back the healthiness of that membrane or recover the peroxidation step of the lipids. If not, then that's the one-way ticket to cell death. The last thing that I want to emphasize on this slide is the instability of these lipid peroxides is a problem. As I mentioned, they become degraded into the 4-NHE and MDA, and those are among the many reactive aldehydes that we see, but these are very well studied for triggering signaling pathways that cause cell death. Okay, this is at the level of lipids. Okay, so ROS-induced lipid peroxidation, and then lipids now are compromised. We have lipid hydroperoxides, and then those degrade. So if we look at this slide here, which is very similar to what we have in the previous two slides, it's adding one more component. So the membrane become compromised because the lipids are oxidized. But then this causes fragmentation. So there is a possibility that this membrane will break because these lipids are now truncated. And since they are truncated, this is probably a possibility where these lipids can form reactions with each other. And since they are hydrophobic, probably can form bleps or micelles, which could make this membrane break. The other component is those electrophiles. These electrophiles, the free electrophiles, are in principle those reactive aldehydes and other components that now will start target other components of the cell. So they can target the mitochondria or other organelles. So we started with an issue with the lipids, but now the issue is spread out all over the cell because it affects the membranous part of all these organs, subcellular organelles, as well as the membrane of the cell. Okay, but now this is within one cell. These electrophiles, in principle, could also migrate and affect other neighboring cells, right? So the issue is propagating more and more. But within the cell, if we want to look at the most known threat, is that when these electrophiles within that cell will attack proteins. And imagine that this protein is a transcription factor. Now you added an electrophile to it, most probably you could deactivate it. So it's not gonna induce anymore its function. Now make it a little more complicated scenario that this protein is actually a transcription factor, and that transcription factor, it induces the antioxidant responses like the enzymes that scavenger those reactive oxygen species. So now if you block it, you can't get rid of those components. So it affects the cell at the transcriptional level. It affects the cell at the membranous level, integrity. It affects the organelles, the structures at their membranes, as well as the flying proteins all over within the cell. Now, let's move forward to to a protein that probably is inserted to a membrane. So this protein is most probably a receptor or a receptor-related protein, so it receives signals. Now, that protein is bound by these electrophiles. So definitely this protein will change the structure. So the third or fourth dimension of that protein is going to be compromised. And then this will lead to the protein damage. So that's what we call protein adducts. And this is a concept I really want you to remember because it's important for you to understand that these intracellular aspects will be in the next two slides transferred and in other words translated into an aspect that is signaled toward other cells and could cause signaling to propagate across the tissue all over. So what is the protein adducts and how the lipid peroxidation causes those? It's very simple. Exactly what I just explained here. But now it's more elaborated here. So you have a damage, you have an induction of oxidative stress and ROS. So now if you look at these phospholipids here, you have oxidative process that happened on those, and these are peroxidized. It happened that on that membrane, you have a protein that is bound to the membrane, very similar to what is explained here. So this is a membrane is embedded within a protein embedded within the membrane. Now this protein is being attached to these phospholipids, but now you can already tell that the healthiness of these phospholipids is not the same. They are compromised. You can see in blue that those are kind of truncated, and some of them are peroxidized. In addition, those broken products now generate the lipid peroxidation products, the aldehydes and the truncated electrophiles. So these truncated electrophiles will form a complex with that protein, which is here in this red. Now that protein is not functional anymore. So we generated those aldehydes, the 4-NE-MDA. It's again written for you here. Please remember these two names. And then those newly generated lipid peroxidation product will form those protein sided chains that form covalent bonds and stable bonds with the proteins. And that's what we call oxidation-specific epitopes. Oxidation-specific epitopes. Okay? Those epitopes, when you say the word epitope, this means it's related to an antibody. And therefore, an antibody response might take place sometime. It's not ready yet. Just bear with me a couple of minutes. So we generated these proteins and complex them with these peroxidation products that are now called oxidation-specific epitopes. Okay? Now, when we get to the level that the compromised membrane and the formation of these oxidative specific epitopes, we are generating within a cell a component that is normally not generated for physiological functions. And therefore, the oxygen-specific epitopes, modified proteins or lipid, could be sensed as a non-self protein. And therefore, these present a damage response for the cell. And that's where the name comes from. They represent a unique class of damage-associated molecular pattern, DAMPs. So that's how DAMPs are generated within our cells. And if you look at immune responses, which we will be describing in the latest classes, it can be triggered by DAMPs, which are damage-associated molecular responses, and infections, which are toxins or pathogen-related associated molecular pattern. And in that case, we call it uh, uh, what's called there. It's the next slide. DAMPs. Yes, it's in this slide here. So the DAMPs is that component that we generate within the cell by lipid peroxidation. So it's a component of a protein and lipid peroxidation. And then the BAMP is a pathogen-associated molecular response that comes from bacteria or virus or toxins. Okay? So with that, we come back to this slide again. And I hope now you have a very good picture of what has happened in terms of loss of oxygen that causes an effect on the cell because there is less ATP, and then therefore there's a change in the calcium levels, and those calcium levels causes the water to rush in, and then water rushing in causes the dilation. Dilation causes mitochondria change in structure, endoplasmic reticulum change in structure, the detachment of the ribosomes, the loss of the translation of the proteins. And at the later stage, the damage is more related to the lipid peroxidation because we have now rushed a lot of oxygen, and that oxygen will generate the ROS, and that ROS is going to cause lipid peroxidation, which will feed back to an effect on the membrane by changing the phospholipid component, and feedback to the proteins by adducting those proteins and cause the damp generation. Okay. Now these damps are now components that trigger the innate response, the innate immune response. So if you imagine that this cell here died, it will release those damps in the bloodstream or around this extracellular matrices. And therefore, when the damps are released, this will be a trigger for the immune response to send those cells that has to be on site to clarify what's going on and inspect what's going on, where this molecule came from, which cell has been damaged, and if this cell is dying, we have to get rid of it. So the molecular cascade is like still continuous. Probably we're gonna stop it here for the next couple of slides, but this will generate an immune response, and that immune response is gonna be like, I'm just making a snapshot for you here, ROS-mediated neutrophil cell death, because neutrophils are first of all the highest in number, and then they are circulating constantly in our blood because they can detect those damages. So if a neutrophil got into a stimulus, which is endogenous or exogenous, so imagine there's a cell that died here and it send out the damps, or there is a cell that is affected by a toxin or infected by a bacteria or a virus or a drug that causes reactive oxygen species induction. Then this neutrophil will quickly induce the NADPH oxidase, which is on the membrane of those neutrophils. So these neutrophils now putting in check the levels of the oxidative stress because they are producing more reactive oxygen species based on the stimulus they are getting from the next door damaged cell or infected cell. And now it has to evaluate the levels of the reactive oxygen species in order to survive or to die. So it's taken a part of the immune response, and at the same time, it's being affected by that immune response. And the only dictation that it gets is the level of the reactive oxygen species. If the level of the reactive oxygen species is high, in other words, the more the stimulus is high, the more bacteria is around that is killing that cell or virus, the more TNF alpha, which is a cytokine that is released from many of those immune responses, then the more reactive oxygen species that will be generated by NADPH oxide. And therefore, that balance here tells if the cell survives or really go to apoptosis. So that's how most of the time neutrophils die when they get to the site of an injury because they are affected by that stimulus from those neighboring cells and then generate tons of those reactive oxygen species, which leads to the adverse effect that I just explained to you at the level of the membrane and the level of mitochondria, sarcoplasmic reticulum, loss of ATP, peroxidation of the membranes, and loss of integrity. So depending on the level, likely also the localization of the reactive oxygen species by the NADPH oxidase, neutrophil may die due to apoptosis, necrosis, but may also show increased survival, depends on the level there. So that's how a molecular event turns out to a cellular outcome. You could see that this neutrophil arrived there at the site of injury and reacted to that site of injury. So let's take a more complicated scenario, or it's a very recent scenario, if you want to say. So now you have two people we are discussing about, which we are looking at the neutrophil reaction toward the ROS being produced in one individual, which has a very well-fared balance between the production of antioxidants to counterpart the pro-oxidants or reactive oxygen species. And there's another personal here which have an issue in generating enough antioxidants. And therefore, there will be a more accumulative process in this personal in case of a viral infection. Now let's take the scenario that these two personnel, they got COVID-19 infection. Well, in that situation, since we have enough antioxidants, neutrophils will arrive to the site and will cope with the levels of the reactive oxygen species. And therefore, they will have a well-balanced defense. Some of those neutrophils will die, but most of them will survive because the level of the reactive oxygen species is not severe enough to kill them. Remember this one here. However, in this person here, the amount of the antioxidant is not going to be in favor of these neutrophils. And therefore, you will have the scenario where the stimulus is going to induce this neutrophil to generate more of the reactive oxygen species. And you can see as you go down, the more ROS, the more chances it's going to die. And therefore, this person will have hard time fighting against the COVID-19 compared to that one. Now, What could help in this situation is to give antioxidants from exogenous sources, like probably from vitamins and other cofactors like zinc that could enhance the activity of the uh, enzymes like glutathione peroxidases and such. So this is another practical aspect of how do you look to that cellular mechanism that could lead to effect in terms of survival in total. Now, the next step to figure out is that whenever you have these cells are in coping with reactive oxygen species, most probably they will turn on certain signaling pathway that normally is not turned on. So the level of the reactive oxygen species also dictates what signaling pathway is going to be turned on. And I'm describing only one for you here, which is the inflammasome. The name inflammasome uh, is a term that is given to a complex of multiple proteins. And as the name indicate, it would induce an inflammatory response. So if you look at this slide here, the inflammasome is a component that made of first pro-caspases. I will be describing caspases later on, but these are molecular scissors which cut proteins. Pro means inactive caspase, and when it's active, we say caspase. Then apoptosis-associated speck-like protein, which is a protein that is very important for the induction of the apoptotic cell death. And then the NLRP3, again, I'm sorry, I can't make this simple. NLR family pyrene domain containing 3, together with this component here, which is the thyrodoxin interacting protein, this guy. So how does the cascade start, and how did the cell induce the generation of inflammasomes? Just bear with me 1 second. Okay. So as described in the previous slide, we have a trigger, which is the pathogen associating molecular aspect, and probably a toxin or a bacteria, or the damps, which are generated from protein adducts. Those will signal on the membrane by inducing the NADPH oxidase. And that NADPH oxidase induces the generation of ROS. So this is one source of the ROS. A damage signal or a toxin or a bacteria or infection. The second source could be the mitochondria, and we saw that with the electron transport chain. And the third source could be coming from degradation of subcellular organelle. We will describe autophagy at a certain point. So any of these three, when it raises the level of the reactive oxygen species, normally this is sensed by two proteins, the thyrodoxin, TRX, and the thyrodoxin interacting protein, which is TX and IP. As this gets to a high level, this complex normally is being generated and incorporated in the cytoplasm of the cell. When ROS is very high, then this complex is dissociated. And the component TXNIP, which is the thyroidexin interacting protein, now dissociates away. It happens that this is the last part of the key lock of the inflammasome. When you have this free, now it can get incorporated with the apoptosis associated spike protein, with the caspases, and with the NLRP3 proteins. So now this inflammasome, what it does, it makes this procaspase 1 and 12 active. There's a processing that takes place that a cleavage of a small part of it is taken away. And now the caspase 1 and 12 are active. What does it mean? Now caspase 1 and 12 are able to migrate around and find their targets. And it happens that those targets are interleukin 1 beta and interleukin 18. And I'm pretty sure if you had any immunology course before, you know that interleukin 1 beta and interleukin 18 would trigger inflammatory responses. So now the cell, it got a damage response, a damage signal. That damage signal is translated into an induction of reactive oxygen species that is sensed by this complex in association with the NLRP3. We have the generation of the inflammasome, and that inflammasome now activated caspases in order to report out that we have an inflammatory response that has to be taking place. If you take any tissue and you want to check that there is an inflammatory response taking place, you will look at the levels of the interleukin 1 beta. If the interleukin 1 beta are high, this is a very good indication that there's an inflammatory response that is taking place in this tissue. Sometimes the dynamics change, but it's like a clear signature that an inflammatory response is taking place. Interleukin 1 beta goes up. Now, the type of cell death that could be induced, again, I told you these names become more famously changed. So inflammasome activates a type of cell death that's called pyroptosis. In other words, this is an inflammatory response that induces cell death. But it's not necessarily that it only causes pyroptosis. It could be also associated with apoptosis, the programmed cell death, and the necroptosis as well. So again, this is another example how we are turning now from a molecular aspect to a cellular aspect, and then it's a systemic aspect if you look at it. Now this interleukin 1 beta is a cytokine, and therefore this cell can secrete the interleukin 1 beta into the bloodstream, and that is going to be circulating all over the body, and therefore it increases the level of the awareness that there's an inflammatory response is taking place somewhere in the body, and we have to take care of it. And probably the inflammatory response from those neutrophils is not enough. More specialized cells of the immune response has now to come into act because we have encountered induction of reactive oxygen species and generation of secondary signal, which is the interleukin 1 beta now, that is being sensed out to call the rest of the immune response to the show. Okay. I think that's it for the reactive oxygen species and mitochondrial damage and how those relate to cell death. Now I will move a little bit to discuss the types of cell death, and I will remind you with some of the aspects that we have already discussed before. So remember, when we discussed the positive and the negative regulation of the cell death in module 3, and we said that this is purely depending on the cyclin levels and the switch from one to another. Now, when we discussed the negative regulation of the cell cycle, we mentioned that this is depending on few proteins like retinoblastoma, P53, and P21. So these slides are exactly the same. Nothing has been changed. I'm just reminding you about that, but you have to review those to get a good hold on this coming lecture. Now, within this aspect, we described P53 being a tumor suppressor. In other words, it blocks the generation of tumors. And whenever there is an unstressed state, the levels of the P53 are under check and they go to proteasomal degradation. Now, in conditions that there's a stress state or there's a damage in the DNA, a metabolic dysfunction, a hypoxic condition, an oxidative stress, a nutrient deprivation. So all that we already have covered now. Or whether there is an oncogene expression, which means a virus has, for example, infected and caused an induction of an oncogene. All that will cause the phosphorylation events of the P53, which now becomes more stable. And P53 will induce some downstream targets. Remember, I mentioned there's many effects of the P53. One of them, P53 induces apoptosis in non-transformed cells, mostly by direct transcriptional activation of the pro-apoptotic PHA3 only proteins, PUMA and NOXA. Okay, so this is the link or one of the links between a damage related to the cell, either from DNA or all these aspects that I described, to an activation of the P53, or other words, stabilization of the P53, and then the induction of these pro-apoptotic proteins. So now I want you to remember that this is going to create a shift in the balance of these pro-apoptotic proteins, which are normally generated in an equal level to the anti-apoptotic proteins. And that's exactly where we are heading in the next few slides. Okay, so there's 3-4 slides. I'm not going to go to the details of these because we already described them extensively. Like we mentioned, if there is a mutation in the P53, how that would affect the function. And more importantly, this is very well correlated to the survival of the cell, because if there is any damage in the DNA, then it's better that this cell stops dividing rather than continue dividing with an error. And that's why we will induce the cell arrest. But at the same time, we have to get prepared in order to get rid of that cell in conditions that we could not resolve the issue or fix the issues that had taken place in the protein, in the DNA damage. And the cell had prepared itself, as I said. Now it start inducing those proteins. It start producing the two pro-apoptotic proteins. So if this happens, then P53 now is the central player which blocks the cell cycle and induces the cell death. And that induction is in 2 levels. First, it induces the P21, which affects the cyclins. Go back to that lecture, affects the cyclin and blocks the cell cycle. Then PAX and BUMA are upregulated, and now those will lead to the cell death. Okay, so here I want to introduce for you the BCL2 family. The BCL2 family, these are groups of proteins. It's a family of proteins broadly divided into 3 different groups. Now, I don't want you to remember those domains, but I really want you to remember the category of those BCL2 families. In this one here, it's what we described with the P53, that if the cell has sensed any of these damages, it will start producing the pro-apoptotic PH3 only proteins, PUMA and NOXA. Okay? Yes, these are the very well known, but we don't want to underestimate any of these others, like BAD, BID, and truncated BID, and all those being. So the BH3 family proteins of BCL2, they all have a common domain that's the BH3. But in terms of function, you can divide them into 2 groups, the pro-apoptotic effector proteins and the pro-apoptotic BH3 only proteins, because they don't have all these other domains. Now, these are counterparted by the pro-survival BCL2-like proteins, the secondary group here. In other words, our cells will generate fairly equal amount of these pro and anti-apoptotic proteins, by which the pro-survival proteins sequester the pro-apoptotic protein and their effectors. Okay, so that's a normal homeostatic situation. But this is an indication that the cell is ready to shift toward death simply by producing more of these pro-apoptotic and these 2 groups, pro-apoptotic proteins. Then the molecular levels will counterpart or exceed the pro-survival BCL2 proteins. And that's exactly what happened in the scenario that I just described for you here, because whatever that signal that causes the damage and induced P53 stabilization, it caused more PUMA and BLAX generation. And therefore now you are producing more of that group here, which not necessarily will have counterparts to sequester in terms of the pro-survival BCL2 proteins. And then this will be a trigger to cause these pro-apoptotic proteins to induce cell death. So that's how I try to link for you molecular aspects from an injury level toward cell death. Now, in order to execute cell death, we need other proteins to come to the show. Whatever type of cell death, we need other proteins to come to the show. What are those? These are the caspases. The caspases, as their name indicate, again, try to remember why we call them caspases. So I wrote it here for you. So these are cysteine proteases that cleave after an aspartate acid residue. So C is for the cysteine and ASP is for the aspartate. And ases means it cleaves. So these are proteins that can cleave any protein that has a consensus sequence of cysteine and then aspartate, followed by aspartate. Of course, they have different consensus sequences. There are many of them, and now they are divided into 2 groups: the effector groups and the initiator groups. The initiator groups are here, 8, 9, 10, and 2. And then you have the effector groups, which eventually become active and execute the process of the apoptosis. And this is our caspases 3, 7, and 1. And remember, in the previous few slides, I described 1 and 12. because those are described in the inflammasome, and these caspases here are mostly executing the apoptosis and part of the necrosis process. Okay. So then what happens? The cell dies. Now we have a shift in the balance of the BCL2 family. We have a reactive oxygen species, a lipid peroxidation, mitochondrial damage, calcium induction, DNA damage, all that affected integrity of the cell, ATP synthesis, and now we have the effector proteins, BCL2 levels are being shifted, and the last who come to the show is the caspases, which will probably start cleaving those proteins. So apoptosis per se, I think I don't have to define it for you now because you already have the discussions through all the slides till today. It's an active process of self-destruction where the cell has a programmed death, which happens both in physiological and pathological conditions. One of the most known physiological condition is, for example, the separation between our fingers, the digits. Without apoptosis, our hands might not have been separated fingers. So this is a process that takes place during the embryonic development. But yet, with the shift of the balance of an injury or infection, cells go to the apoptotic process. And that's due to a severe injury, accumulation of these misfolded proteins, infections, obstructions of the tissue, or loss of oxygen. So all that can cause the programmed cell death. Now, before going to the definitions and how we can define necrosis versus apoptosis and what happens, probably it's a very good point to go for this video. Let me know if you see my screen.
Apoptosis.
Do you see my screen? If not, please let me know on the mic.
Yeah, we can see it.
Okay, thank you.
Apoptosis extends over 3 steps: initiation, execution, and phagocytosis. Initiation may be initiated by 2 different mechanisms, depending on whether the initiating signal stems from inside or outside the cell.
Okay, doesn't seem that it's working.
Intrinsic pathway of initiation. The most frequent signals that activate intrinsic initiation are irreparable and irreversible DNA lesions. These lesions lead to the activation of ATM, a protein that is able to activate the tumor suppressor protein p53. In addition to many other proteins, p53 is able to activate the protein BAX. BAX forms pores in the outer membrane of mitochondria. Calcium ions, protons, and other molecules which are located in the intermembrane region between the outer and the inner membrane may leak into the cytosol. One of these molecules is the protein cytochrome C. In the cytosol, cytochrome C binds to APAF and may then associate with an inactive form of procaspase 9. As a consequence, the inhibiting domain of the procaspase is hydrolyzed and dissociates from the protease. By this mechanism, caspase 9 becomes active. Active caspase 9 is able to cleave additional caspases and to destroy many other proteins. In this way, caspase 9 initiates the so-called caspase cascade. This cascade denotes a stepwise cascade of proteases that cleave and thus activate each other. The concentration of active proteases in the cell increases rapidly, leading to the destruction of many different proteins. Extrinsic pathway of initiation. The extrinsic pathway of initiation is caused by extracellular signals. A common extrinsic factor that initiates apoptosis is tumor necrosis factor alpha, abbreviated TNF-alpha, which is secreted by many cells, like T killer cells. Such cells can induce apoptosis in cells that are no longer required or are dangerous for the organism, for example, tumor cells. TNF-alpha binds to the TNF receptor at the outer membrane of the cell. Subsequently, the so-called death domain at the cytoplasmic side of the receptor is activated. As a result, cytosolic proteins with their own death domains bind and are activated. The first protein that binds to the cytosolic part of the receptor is the TNF receptor-associated protein with death domain, or TRAD. Next, the protein Fas-associated protein with death domain binds, which recruits procaspase 8. This protease is able to autocatalyze the hydrolysis of its inhibiting segment, leading to active caspase 8, which disassociates from the receptor and is then able to initiate the caspase cascade. Execution cleavage of DNA. Important steps of the execution phase are the cleavage of DNA and the cleavage of the cytoskeleton. In the normal cell, DNase is complex to an inhibitor and is inactive. After initiation and activation of the caspase cascade, the active caspase 3 is able to cleave this inhibitor. Activated DNase cleaves DNA. Cleavage sites are located at regular intervals of 180 base pairs. In between, histone proteins of nucleosomes protect the DNA against DNase cleavage. Execution, cleavage of the cytoskeleton. Caspase 3 cleaves many other proteins, such as proteins of the cytoskeleton. Hereby the cell loses its structure. Next, other proteins cause the cell to collapse into vesicles, the so-called apoptotic blebs. Most blebs contain mitochondria and also portions of the nucleus including DNA. These components allow energy to be maintained and new proteins synthesized. The rapid breakup of the cell and of the formed vesicles is thus avoided, preventing an inflammatory reaction in the surrounding tissue. Phagocytosis. The various processes of the execution phase lead to significant modifications of the structure and composition of the outer membranes of cells and apoptotic blebs. Based on the modified membrane structure, phagocytes, like macrophages, can recognize the blebs. In the cytosol of the phagocytosing cell, the blebs fuse with lysosomes. These organelles contain enzymes that finally metabolize the blebs and their components.
Okay, so this is kind of a very good introduction for the apoptotic processes: intrinsic, extrinsic, the execution process, and then the cleanup that takes place. And that's why we call apoptosis a programmed cell death, because when the cell dies, in principle, it's kind of dismantled into smaller components. And if you hear in the video, it says it doesn't induce any inflammatory response that could propagate to cause damage. The only component of the inflammatory response that is associated with the apoptosis is the engulfing of those small blebs of the cell. The other good thing about apoptosis is that most of these blebs contain cellular parts that could be recycled and could be reused. This is totally opposite to what we see in necrosis. So here, there is a comparison for you between necrosis versus apoptosis. It's very brief. If you want to have more reading, please go to the book at page 85, and you can read the text. Again, many times I'm adding to you later after the class readings like some PDF files that's 1, 2 pages, 3 pages, which gives you more information. Because if you see this video that we have described, it's very condensed and it's through a lot of information. Not necessarily that you are familiar with it. So please go to the readings, because I can't have the capacity to add all the readings in the slides. So what's the comparison between necrosis versus apoptosis? Well, apoptosis was very well described in the video, where it's programmed, it's energy dependent. So the cell, while dying, it still needs some energy to use in order to perform these cleavages and formation of the blaps in a very synchronized and clean fashion. The cell normally shrinks during the apoptotic process, so you compare this size to that size. So in necrotic cells, they become swelling. There is chromatin condensation, and now you can tell why the chromatin condensation, because of the activation of those DNases, which will cleave those DNA within the cell. And then the membrane blabbing is another component where the cells start to break down into much smaller cellular organelle. Like, it is caspase mediated, so caspase cascades has to be induced in apoptosis, while yes, caspases are partially affecting the necrosis, but they are not the major trigger. And it is non-inflammatory. So if a cell dies, it doesn't induce an immune response that takes place against the cell itself. All we need to do at this particular step is just to engulf those bodies, and the inflammatory response stops. There will be no recurrent inflammatory response. While with necrosis, the cell becomes bigger because of the swelling, mostly as I described with the calcium load and the rush of the oxygen, and then the swelling of the endoplasmic reticulum. Then the swelling itself, that's what induces some blebs in the membrane. So it's common, but the swelling is going to cause at later point a rupture of the membrane, not a formation of these apoptotic bodies. And finally, this progressive injury will cause the breakdown. And remember the damps that we described. These damps will be released, and therefore these damps will induce an inflammatory response that will call the immune cells, and most probably will call other immune cells other than the neutrophils to the site. So this is like a brief overview between apoptosis and necrosis. There is a video here that I chose for you, and if you look at the videos that I'm trying to find, unfortunately, we don't have really a good source all the time. So I try to look at Nature, Harvard, very well-reputed institutions which generate these videos. If you go to YouTube, you might find a lot of YouTube channels that are operated by personnel, not necessarily being like trusted. So be careful when you look at these YouTube channels. Okay. Based on our time, we still have like 20 minutes. I might go, let me see how many slides I still have. So probably I will go till here. So I'll go till here, and then we start metabolic diseases next round. So this is just a recap on the apoptotic signaling pathways, that it's divided into intrinsic or extrinsic pathway. Again, what I want to emphasize is the fact that the BCL2 family anti-apoptotic and pro-apoptotic proteins are in fact the major trigger of the intrinsic signaling pathway. We described also in previous slides the cytochrome C release. So what happens in a situation where you have an intrinsic pathway is induced, like DNA damage? Like we go back here, now you can have a global overview of what we have described over 3 or 4 lectures. where whatever the reason, it's a ROS related or whatever damage related to the DNA induces p53, and p53 induces the level of NOXA and PUMA. Okay. Now, NOXA and PUMA are pro-apoptotic proteins. So the blue proteins are gonna be overcoming the red anti-apoptotic proteins. On top of this, you can see that PUMA and NOXA could block some pro-apoptotic proteins. So they block their function. And if we have a lot of these being generated, then the backs and back, so I will go back a little bit to that slide. So remember, PUMA and NOXA are pH3 only. These are, we described them from the p53 arm. And then the backs and back, these are the pro-apoptotic effectors. But now these pro-apoptotic effectors are normally sequestered with the BCL2 that you can see in the slides here. So the BCL2 and the BCLXL, BCLW, and MCL1, those sequester the backs and back at the mitochondrial level. Now, when you have a trigger that induces the levels of BIM, BED, and BAD, which are the pro-apoptotic proteins, backs and back now can be released because you have less pro-apoptotic proteins available. And those will go to a process called oligomerization. In other words, they get recruited into the mitochondrial membrane. Okay. This already tells you that there will be a compromise for the mitochondrial membrane in terms of ATP and in terms of the integrity of what is getting in and what's going out of the mitochondria. The other factor is the cytochrome C and the SMAC. So these are two different proteins that are released from the mitochondria during the cell death. Now, what happened when those are normally in the mitochondria and released into the cytoplasm? They will be recognized by the procaspase 9. Okay? So together will form what we call the apoptosome. And now, when exactly as we described before for the caspase 8 and the caspase 112, when apoptosis is induced, then the procaspase 8, the APAF, and the cytochrome together leads to that procaspase to become an active caspase. That's what was described in the video. And then it says it could cleave other proteins and it could cleave caspases 3 and 7. So it activates the caspase cascade activation. Now caspase 3 and 7 will start cleaving the rest of the proteins. The other arm was the extrinsic pathway, where you have a death signal, tumor necrosis factor, Fas ligand, trail, and whatever that ligand is. It binds to a death receptor, and then the complex starts to form TNF alpha receptor related death proteins, RIP kinase, FAT, caspase 8, all those form an active caspase. So the cascades that you are seeing is almost similar. The only change is the type of the proteins that are being recruited, because here you need cytochrome C, you need the APF1, and you would need procaspase to generate that apoptosome. On the membrane, you will need the TRADD, which is TNF alpha receptor associated death domain, because now this protein is specialized to bind to the inner part of that receptor. And then RIP kinase 1 and Fas ligand associated death domain will form this complex. But what this complex do? It activates caspase 8. And what this component do? It activates caspase 9. And now both caspase 8 and 9 induces the active caspases 3 and 7, which execute the cell death. More information are written for you here in terms of text. If you read it, it will be much clearer. And also it's presented in a different way for you here, where it's showing you the balance between the BCL and the pro and anti-apoptotic proteins. So out of these, I just hope that you go through it till next week and let me know if you have any questions regarding for the apoptotic signaling pathways in comparison to necrosis. And if there's anything not clear, you let me know. Of course, there is a crosstalk between the two. It's something we have to acknowledge, that extrinsic pathway, when caspase 8 is active, so you can see here it's presented in a different way. Caspase 8 and 10, cause caspase and 10 are active. They will cleave BID to become a truncated BID. And if we go back into that slide, you will see truncated BID as a pro-apoptotic protein here. So that's a link between extrinsic and intrinsic pathway. Now the other common link is that caspases 8 and 10, as well as caspase 9 from the apoptosome, they both lead to the activation of caspase 3 and 7. And in the same, they take the lead in terms of the apoptotic process. Now, I'm sure there will be a question. Do we need to know the name of those molecules? BAC BCL2, as well as cytochrome C, APAF, apoptosome, inflammasome. I need you to know what are the components. Okay, I'm not gonna ask you what is BAC, B-A-K. Can you write what is BAC? I'm not gonna ask you what is that because it's a BH3 member, and but in terms of function, I will ask you what is the function. Okay, and here I just add for you how complex this domain is. Cell death is indirectly related to cancer studies. In other words, if we know how to kill cells specifically, we could eliminate cancer. So there's a lot of research about how to eliminate cancer, and could be through a specific cell death that we can eliminate cancer. And that's why you will be always finding in these papers and reviews the fadosome. The fadosome is, in fact, what I just explained for you. You have a Fas ligand that binds to a Fas receptor, and then the death domain and the RIP kinase and caspase 8. So that's the fadosome. The inflammasome, the caspase 1, the ASC, the NLRP3, I described this before when we spoke about the inflammasome. I need you to know inflammasome and apoptosome. These two components, you need to know which proteins make it. But in this paper, if you go, you will see much more than those complexes. And again, it's not really something very complicated. It's simply the types of the proteins that are associated to cause the generation of that inflammasome. And then which caspase become active in this condition. It's caspase 1, which leads to interleukin 1 beta and interleukin 18 induction, cleavage. And in this condition, it's active caspase active caspase 3 and active caspase 7. Okay, so we stop at this point. we start describing the cell signaling, because now we kind of touch on the cell signaling here, that the cell receives a death signal and responds by cell death. In the next slides, I have, so probably I will take them off and I will upload a new PDF for you. How the cell responds to a signal, if this signal comes from a hormone, and then we will start describing hormones like insulin and how it regulates glucose levels and diabetes and all that. Okay, with this, so I'm stopping here. This will be the last slide for the session. I hope you go through it as soon as possible, so we are on the same page, and we'll catch up next week. I'm gonna stop recording right now. And if you guys have any questions, let me know.